All right, so in this video, I'm going to be going over cell respiration, which is chapter 9. And this is going to be kind of a relatively uh, lengthy video in the terms of we're covering everything. We're not breaking it up into glycolysis. I'm just going to kind of go through all of it. So it is all in chapter 9. So this is basically saying what we have to make sure you know from the College Board. You got to make sure you understand cell respiration. Eukaryotes is involved coordinated enzyme catalyzed reactions. We got to cover a little bit of fermentation. We got to know the electron transport chain and really all the ins and outs that happen there. So in terms of the big thing that we're getting into today is why do we care about this? What is the quote unquote energy of life? So you got to understand at least what we get into first, which is going to be making ATP is not going to be really a one-step thing. So I've talked about this in other videos where when we think of this word metabolism, it's all of the chemical reactions in an organism's body. When you think about a metabolic pathway, that means you're going to be starting with one molecule and end with a product, and it takes multiple steps, right? So it's going to take multiple steps to get your end product. And this is showing you that if I had A mixed with B, or excuse me, A makes B, B makes C, C makes D. Each one of them involves their own specific enzyme. So in terms of enzymes, you want to know that A is specific to using enzyme 1 to make product B. B is specific to enzyme 2 to make product C. C is specific to enzyme 3 to make product D. Really common AP test questions would be, well, what if enzyme 3 is denatured or it's mutated, it's not functioning? Would you ever get this product D? The answer would be no, just because that enzyme is no longer functioning. You would still get a high concentration of C, but regardless, you just want to understand what is a metabolic pathway. It just basically means you're starting with one molecule, you're ending with a product that's different, and it takes multiple steps to get there. So that's kind of where I wanted to start off this video, because cell respiration in its entirety is a metabolic pathway. So let's kind of get into the overview first, which is a really big topic that you need to make sure you feel relatively comfortable with. And that's going to be, when you look at the product of photosynthesis, those are going to be the reactants of cell respiration and vice versa. So basically, you're going to get light energy coming in through the chloroplast. You're going to make your organic molecules. And again, organic molecules, that would be essentially glucose with oxygen is going to go into the mitochondria, and then you're going to produce your byproduct of carbon dioxide and water. Those are going to be the reactants of photosynthesis. So it's this really nice cyclical process. So in terms of that, we just kind of got to understand this video about cell respiration. What is the point? What's the purpose of it? Okay. I just wanted to recap on this video again. Putting a phosphate onto this ADP, adenosine diphosphate, right? There's these two phosphates here. It doesn't necessarily naturally want to do that. So cell respiration in its whole entirety makes this whole process actually worth it, in the sense that it makes it not so endergonic. It's actually going to be a spontaneous reaction. So that's where things get interesting. So with this video, we do have to get into a little bit of chemistry, not difficult chemistry, but we have to talk a little bit about these oxidation and reduction. Now, the biggest thing that I'm going to suggest that you guys at least understand is oil rig, okay? Oxidation is losing electrons, reduction is gaining. So basically, when you look at this molecule A, I have these two electrons. I would say that A essentially is losing those electrons. So I would say that molecule A is oxidized, and that's what it tells you here, because it's lost electrons. B is going to be gaining those electrons, so I would say it's going to be reduced, okay? So this big thing here that I like to add to my notes is oxygen is very, very, very electromagnetic. Makes it a good oxidizing agent in the sense that it wants to pull these electrons off, all right? So I'm just going to advise that for you guys, when you study cell respiration, you need to follow, you absolutely have to follow your electrons, okay? Understand where these electrons are coming from, understand where these electrons are going, and that's going to be your best kind of starting point on understanding cell respiration. because when you follow the electrons, this whole process becomes a lot easier. So let's just take a look here at the overall equation of cell respiration. I would make sure that you have this thing memorized, or at least in the back of your mind when you're going through any respiration problems. So first and foremost, you got to know C6H12O6, that is glucose. Don't forget that. You're going to combine it with oxygen. You're going to make CO2 and water, okay? But the biggest thing is that glucose basically has the electrons to give. So when you think about it, oxidation is losing electrons, so we're going to say that glucose becomes oxidized. It loses those electrons, and then it becomes CO2. Well, oxygen, like I mentioned here, it's really, really electronegative, which really just means it wants these electrons. Oxygen is going to be picking up the electrons from glucose, then it is going to form water, and then in the process, you're going to be making energy. Not, I shouldn't say making energy, but you're actually going to be making ATP, which is a form of usable energy. So really, the power is in the electrons. So I can't emphasize that enough. You have to follow where these electrons are going, and then it makes this whole process quite a bit easier. So cell respiration overview. This is the overall equation, and it's really broken up into 3 massive steps. The first one is glycolysis, also known as glycolysis. Okay, I like to say that, that interesting way, because lysis means to split. And this is when you're going to take glucose into pyruvate. This occurs in the cytosol, so basically outside of the mitochondria. We're going to have a citric acid cycle, also known as the Krebs cycle. So you're going to take pyruvate and form acetyl-CoA, and that takes place in the mitochondrial matrix, so we're actually inside. And then the electron transport chain is going to be in the inner mitochondrial membrane, and that's going to be the big, big, big payoff. So let's kind of take a look here. This inside the mitochondria, and this is going to be called the matrix. Again, outside the mitochondria is the cytosol. And don't forget that this whole thing is wrapped up if it is a eukaryotic cell, which is going to be because it has a membrane-bound organelle. Don't forget there's things like the nucleus over here. There's all kinds of other stuff that are happening in the cell. But in terms of this, you got your cytosol. Glucose is what you're going to be starting off with. You're going to convert that into pyruvate. You're going to make some ATP. And then we're going to have what's called the link reaction here. You're going to make acetyl-CoA. Acetyl-CoA is going to join the citric acid cycle. You're going to make a little bit of ATP here via substrate level phosphorylation. I'll talk about that in a second. And then the big payoff is going to be this last step, which is known as oxidative phosphorylation. So, like I mentioned at the beginning, this video is all about glucose. Glucose starting out to make this ATP via substrate level oxidative phosphorylation. But you can have other things like proteins can jump in here, as well as lipids. So, and you think about it, it makes sense. You eat protein, you eat lipids, you can get energy from them. So, granted, most of this video is going to be talking about glucose, but there's other macromolecules that can jump in. So, before we do that, let's talk about this term, substrate level phosphorylation, because this is something that a lot of students get tripped up on. And they think that, oh, it makes ATP. What's the difference between substrate level oxidative? Well, quite a bit. Let's explain. So substrate level phosphorylation, my definition is that ATP is produced, but it's not efficient because you're taking a phosphate, you're removing it from a substrate. So the biggest thing is that it's not efficient. It's a way to make some ATP, but it's not the best made. It's not the best way. So basically what I'm showing you here is that if I have a substrate that has a phosphate, I can move that phosphate over to ADP and making ATP. So did I make ATP? Yes, I did. But it took an enzyme to do that, and it's pretty slow. It's not the best way for a cell to make ATP, but it is a way for some ATP to be made. So with that, I'm going to skip over. I need to talk about it now. So let's get into this. Just to remind you, substrate level phosphorylation. You're going to be pulling off a phosphate. You do make ATP, but it's not the most efficient. Oxidative phosphorylation is by and large the best. So making this thing possible, we got to talk about NAD, NAD plus to NADH. We don't need to get crazy specific here, but basically this is your organic molecule down here. It kind of looks like this cartoonish thing with the electrons, but let's just say that this is glucose here. And basically, electrons have a negative charge. This is NAD plus, okay? Now what you need to understand here is that NAD plus is a coenzyme. It's going to be carrying electrons. It has a positive charge just naturally. Okay, this is what it's showing you. When it's in its essential oxidized state, it has just an overall positive charge. Well, let's just say that if an electron was transferred over, this negative charge and this positive charge would essentially cancel each other out. But when you look at it, you're actually gaining a second electron. So rather than being NAD, well, let me just show you this. If I actually made it in terms of this, it was NAD. I had this positive charge. I added one electron.